Our next panel invites leading drug developers to critically examine the latest advances of this genetic disorder. Let's dive deeper into the conversation as we explore the new directions in Fragile X syndrome therapeutics and ask, how soon can we have the first effective treatment?
Well, welcome to the Therapeutics panel on this Fragile X webinar. My name is Rich Saul, and I'm the Advisor for Strategic Initiatives and head of the Boston office for WuXi AppTec. The Fragile X is an X-linked neurodevelopmental disorder caused by the absence or reduction of the FMRP1 gene product known as Fragile X mental retardation protein 1. The syndrome is most frequent monogenic form of intellectual disability and autism. The considerable efforts have focused on rescuing the synaptic plasticity that's dependent upon protein synthesis, and two of the targets most studied are the mGlu5R antagonist and GABA B agonist in particular. However, no drug approval has been achieved yet based on these mechanisms. Now, a number of innovative companies are moving forward to explore new mechanistic approaches combined with ways to improve trial execution and streamline regulatory processes. And with me today are some of these innovation leaders. Mark Baer is professor of neuroscience at the Picower Institute for Learning and Memory at MIT, and his lab has been studying the role of Gsk alpha in Fragile X. Mark Gurney is chairman and chief executive officer at Therapeutics, a company that, in the case of Fragile X, is examining connections between neurons that don't mature the way they should using phosphodiesterase subtype inhibitors for Fragile X therapies. Neil Thompson is the chief scientific officer at Helix. It's a company exploring AI-powered, patient-inspired treatments for rare diseases. And Walter Kaufman is the chief medical officer at Anavex Life Sciences. Anavex is examining the signaling receptor agonism for the treatment of Fragile X syndrome. And finally, Charles Large, the chief executive officer at Tiffany Therapeutics, said the company is examining ion channel modulators for Fragile X syndrome treatment. I'd like to start with Mark. And how has our understanding of the core pathophysiology changed over the last 10 to 20 years?
Well, when I joined the field in the late 90s, I would say the situation was that the gene had been identified, some of the functions of the protein had been discerned, an animal model was available which was showing some changes in neuronal structure and also in the status of inhibition in the brain. And so that was sort of the state at the time. With respect to developing medicines, I don't think there was much hope that a medical treatment could be devised that would correct core pathophysiology of the disease. Rather, we were confronted with symptoms of the disease like irritability and anxiety that could be potentially controlled or ameliorated in a palliative way. I think the picture changed quite dramatically early in the noughts, 2002 and onward, with the discovery that many of the features of Fragile X syndrome could be accounted for by altered regulation of protein synthesis in neurons. That was one side of the coin. The other side of the coin was that there were specific neurotransmitter receptors and signaling mechanisms that control neuronal protein synthesis in a significant way. And so suddenly we had the prospect that we could develop a disease-modifying treatment based on restoring the normal balance of protein synthesis in the brains of those affected with Fragile X. This idea initially, of course, was wildly speculative, but it was quickly validated in animal studies, both using the mouse model of Fragile X and also zebrafish models and fruit fly models of Fragile X, showing an evolutionary conservation of this mechanism. So it was this insight that led to a lot of efforts in translating to the clinic, particularly developing the mGluR5 receptor inhibitors, mGluR5 being the glutamate receptor that triggers protein synthesis in neurons. As this story was progressing, we were beginning to fill in some of the other key functions of the FMRP protein. So it not only is a regulator of protein synthesis, but it also through direct protein-protein interactions can regulate the excitability of neurons through modulation of ion channels. And it's also, of course, been recognized for a long time that brain development in the absence of FMRP can alter the wiring of the brain such that the ratio of excitation to inhibition is altered, it's increased. So this gave a couple of different paths that one could take to develop disease-modifying therapies. One being specific modulation of ion channels that are affected by the loss of FMRP. And the other is more of a general approach of rebalancing this ratio of excitation to inhibition. So there's been a lot of progress over the last 20 years, and I think we have a lot of shots on goal.
It's wonderful. So, Walter, what are some of the key learnings that are informing our approach to Factor X today?
So if we cover the same period of last 20 years, on one hand, the clinical field was expanding the knowledge about the clinical phenotype and the genetics, particularly with focus on diagnosis of Fragile X syndrome. The first national history study was developed. So from a very compartmentalized approach to studying the phenotype from the perspective of autism, ADHD was much more integrated. And at the same time, because the trials were ongoing, there was a period of evaluation of all the components which are needed for developing new therapeutics. Assessment of outcome measures, biomarkers, assessment of quality of life, specifically in Fragile X, and what the caregivers and the families really are expecting from these therapies. But as more information came, it became even more clear that in this monogenic disorder, the story is complicated. Even though it's a single gene disorder, the phenotypical variability is a major issue. And even if you look at more discrete groups within the syndrome, for instance, those with more severe cognitive impairment who usually have at the same time behavioral problems, including autism and hyperarousal and other behavioral problems like disruptive behavior, that group is not going to present all these symptoms the same way. So on one hand, we cannot separate so well cognition from behavior. Individual variability is a major issue. So there are challenges of this increased knowledge. Most recently, in the last 2 or 3 years, we have come to be better at doing remote assessments, which is actually was enhanced by the COVID-19 situation. So remote assessment, even remote intervention have become a suitable approach. So I think that in the last decade in particular, we have set in the stage for additional treatment development.
There was a question from the audience that we received regarding the future of mGluR5 antagonists, and should we be more, let's say, aggressive about targeting this work at younger ages, or enriching samples for methylation status, something of that nature?
Well, with regard to that, there is still an ongoing trial which is re-examining one of the mGluR5 antagonists that was tried, a few years ago now in a younger population, because as it will be clearly understood, for a developmental disorder, the earlier the intervention, the more likely to have an impact. So that trial is ongoing, is about to finish, and interestingly, that trial is also addressing a number of issues with potential implication for multiple other drug development and multiple other therapies, such as testing of outcome measures, and biomarkers and combination therapies. Because it's that particular study is a combination of a language intervention and drug treatment. So I think that people have to stay tuned. We have to see what is the outcome of that. Now, with regard to methylation, which started with mTOR5 antagonists, it keeps coming back as a story, and it's a puzzling one for many of us because, as you know, only about 60% of those with Fragile X has a complete methylation in blood cells, essentially lymphocytes. So we don't know why those who have complete methylation are more responsive to some intervention. I think that if it initially was doubt to be relevant or not, or a single random finding, I think that we need to be examined in the future I will say in most clinical trials.
Well, thank you, Walter. I'd like to ask Mark Gurney, the recent clinical trials with PDE4 subtype D antagonists have provided incredible insights. And can you share those results and your excitement about the breakthroughs that seem to be making a key difference with respect to outcome measures versus previous trials?
Thanks, Richard. One of the core changes in people with Fragile X is profound intellectual disability. Men are affected more than women. Men have a single X chromosome, and when that FMRP1 gene is inactivated, there's profound changes in brain development. Women are less affected as they have two X chromosomes and they're mosaic in most of the tissues in their body. So they can have a relatively normal brain development if the mutant chromosome is inactivated and the wild type chromosome is active, and profound intellectual disability if the opposite is true, if the good chromosome is inactivated and the mutant chromosome is active. So changes, a decrement in intellectual disability is a core feature of the disease. In males, the average IQ is 40 to 50, and this is limiting in terms of the ability of the patient to live independently or even to live with assistance in an institutional setting. So one of the initial biochemical changes that was reported in patients with Fragile X was a change in cyclic AMP, a decrease in cyclic AMP. And cyclic AMP is an important signaling molecule in pathways for neuroplasticity that allow neurons to make connections and store both short-term and long-term memories. So the initial observation of the decrease in cyclic AMP was made by Dr. Elizabeth Berry-Kravis, who's now a professor of pediatric neurology at Rush University Medical Center in Chicago. And what Dr. Berry-Kravis showed was that cyclic AMP was decreased in the tissues that she sampled. And she showed that prior to the identification and cloning of the FMR1 gene. So this is a bedside to bench translational program, because after the gene was cloned and the Drosophila, the fruit fly and mouse models of Fragile X were developed, it was found that there was a decrease in cyclic AMP in both of those animal models. So we've developed a compound, BPN 14770, that targets a phosphodiesterase known as PDE4D, which increases cyclic AMP in the brain. We know that PDE4D is an important component of learning and memory in humans because ultra-rare missense mutations in that gene cause profound intellectual disability. The disorder is known as acrodyssystosis type 1. So having that in hand, we were following the literature, and there's been considerable work on whether or not modulation of cyclic AMP in Drosophila and mice might improve the Fragile X disorder in these models. So we collaborated with the FRAXA Foundation. They performed preclinical testing in the FMR1 knockout mice. And what we found is that our compound improved behavioral phenotypes. It facilitated maturation of cortical circuitry. And when you washed the drug out and retested the mice 2 weeks later, the behavioral improvement was maintained. So that provided us impetus to go into a phase 2 clinical trial with our compound. And lo and behold, the compound or the clinical trial reported positive findings. So we used, for the first time, a set of cognitive tools that were developed as part of the NIH toolbox. So these are cognitive tests that can be performed by people with profound intellectual disability. So we were directly assessing cognitive performance over the course of the clinical trial in adult men with Fragile X. We also incorporated caregiver measures, a visual analog scale that assessed 3 different domains of patient function, as well as a physician-reported cognitive scale or outcomes. And what we found is that using the NIH toolbox, we saw improvement in in test assessing verbal knowledge. Patients and caregivers in their reports assessed 3 domains, 2 of which were language and daily function, and both of those were also found to improve. We did not correct for multiplicity of testing. This was an exploratory trial, and we had a large number of different assessments that we conducted. Nonetheless, we're very encouraged. We have a drug which in mice affects cortical circuitry. We expected a change in cognitive function, and that's exactly what we're seeing. We also saw trends, and we also saw persistence of effect. So this was a crossover trial. In the first arm, people on drug did better than placebo. In the second arm, the people that started on drug and crossed over to placebo showed continued improvement or maintenance of improvement. There are also trends for improvement in biomarkers. We saw a trend for improvement in the amplitude of the auditory evoked response potential. And we also saw improvement in a biomarker that's altered in people with autism. So we're really encouraged. We used a novel clinical assessment tool, the NIH toolbox. This is something that has not been presented to FDA prior to our trial. So we're working with FDA now to help them understand the clinical significance.
Well, thank you. They're very exciting results so far, and I wish you luck in the later stage trials. So let's. I want to come back to a question around the animal models, but we'll come back to that a little bit later. I'd like to move on to the question about, why should we be as optimistic about other targets or pathways that could be of significance in treating Fragile X? I'd like to start with Charles, if that's possible.
Yeah, sure. Thanks, Richard. So I think a lot of the optimism really comes from what's been said already in this call. We've understood a huge amount more about what FMRP does and how it regulates activity in neurons and how it helps those neurons to develop. And in fact, one particular area that was mentioned earlier was in relation to ion channels, where we understand at quite enormous detail now how the FMR protein can bind to specific ion channels. And by binding to those ion channel proteins, it then acts as a point of contact between the activity of the cell and translational regulation, which is, of course, its main job of the protein. So it's a wonderfully exquisite way in which the neuron can combine activity of the cell, excitability and so forth, synaptic activity, with long-term plasticity, with the ability of that cell to make new proteins, to develop synapses, develop dendrites and so forth. So I think this relationship with ion channels at the center of it is leading to a number of quite exciting opportunities for modulating this pathway in the reduced context of FMRP. So where FMRP is reduced in the illness, there's the opportunity to try to compensate for that with treatments that are targeting particular ion channels. And I think what excites me, who's worked in ion channels for a very long time, is that ion channels are very tractable drug targets. So you can interact with an ion channel, and by doing so, you can alter the activity of a neuron. And that leads to reasonably immediate effects on a circuit, which can lead to effects that you can pick up with some of the methods and biomarkers that have been mentioned already, like EEG or auditory evoked potentials. So I think what we're seeing in the field, and this isn't just for Fragile X, I think we're seeing this across neuropsychiatry, is the ability to jump backwards and forwards between some of the animal models and looking at neurons, looking at circuits, and then jump forward into patients and look at biomarkers linked to electrophysiology, such as EEG, fMRI, transcranial magnetic stimulation combined with EEG, and so forth. One area in particular that we've been looking at, which we think is quite exciting as well, which is quite particular, I think, for Fragile X, is changes in the sensory system. So hyperacusis is a common symptom that's seen in people with Fragile X syndrome. So this hypersensitivity to sound. And you can trace that back to particular pathways in the auditory brainstem and how specific ion channels, in particular the Kv3 ion channel, is affected by the reduced presence of the FMR protein. So once again, you can go backwards and forwards between the animal models and looking at the impact of the gene at the local neuron level, all the way through to understanding how a particular symptom cluster is affected and produced in the patients. And so we're very excited, I think, about the translatability of some of these novel targets where you can influence these pathways and hopefully influence the symptoms behind them.
So Neil, how is AI being used to elucidate new pathways, new targets, or maybe looking more closely at the data, let's say?
Yeah, thank you, Richard. So I think the discussion so far today has really highlighted how even a single gene defect can lead to a very complex biology. And it's great that we have some identified targets that we can test preclinically and now clinically, and we're really hopeful that they'll bring some benefit to patients. I think, though, if we're realistic, the expectation would be that we probably need to target several mechanisms if we are going to treat all of the symptoms in all of the patients. And that's when it gets quite complex. Sometimes the knowledge that we have allows us to select drug mechanisms that can do that, can target several pathways or processes. However, it can get quite complex. So how do we do that? And the answer, I think, is within larger data sets, for example, gene expression data or proteomic data. That more detailed analysis of the changes that take place within a disease situation in contrast to a normal cell type. And it's at that level that AI really comes to the fore. It allows us to look for those patterns and differences and the connections between the changes and potential pathways that may be new and unthought of yet as relevant to the disease. And that's what we're seeing. We're seeing the ability to analyze this data and to identify new mechanisms and ultimately new targets that may be able to come forward and provide this way of targeting several processes. The other way, of course, is to use several drugs at the same time, a combination of drugs, which is very often the first clinical approach when drugs become available for patients. And again, I think it's possible to see there that the analysis by machine learning can allow you to evaluate the potential beneficial or possible not beneficial effects of particular drug combinations and select those that may target the particular disease mechanisms of interest in this disease. And we're already seeing that coming through with some new mechanisms and potential combinations to target those. So I think those are two main areas that can easily lead to some new ideas and new molecules that can be tested here. But in fact, I think AI has the potential to impact the drug discovery and development at many levels later on in the process as well, in terms of, for example, equating animal model phenotypes with human phenotypes and providing a better link and understanding there too. So lots of potential.
Great. Mark, you've been studying Gsk3 in particular, alpha in particular, and what kind of insights are you learning from that? Mark Baer.
The Gsk3 work is really a continuation of the work we've been pursuing for 20 years on identifying nodes in the signaling pathway that couple the mGluR5 receptor or glutamate release to FMRP regulated protein synthesis. So we've been working our way down the path inside the cell and looking at key regulators. Gsk3 happens to be one of those. This is an enzyme that has attracted a lot of interest for many years because it's believed to be the target of lithium, which is, of course, an effective therapy for bipolar disorder. So there's been a lot of interest in developing Gsk3 inhibitors. Unfortunately, the selective inhibitors that have been developed have the terrible side effect of promoting tumor growth. So most pharmaceutical companies abandoned their Gsk3 programs some years back. But in the meantime, it's been discovered that, well, I guess we've known for a long time that there are two paralogs of Gsk3, alpha and beta. And only until recently has it been possible to target just one of those paralogs at the exclusion of the other. And these experiments have shown that you can still get a therapeutic effect of inhibiting one of the paralogs without causing any cancerous growth. So it's a dawning of a new day in terms of Gsk3 biology. And one of the paralogs in particular seems to be playing a central role in the pathogenesis of Fragile X.
There's exciting developments, that's for sure. We've been discussing small molecules for the most part during the last few minutes, but Walter, what might we see with genetic approaches, either gene therapy, associated viral vectors, ASO? Do we have to worry about drug delivery into using these regimens?
Well, historically, gene therapy actually was an approach that was investigated shortly after the identification of the gene in the 90s. And Fragile X has a unique situation. It's a single gene disorder, and the gene is the coding region is normal. So the problem is with regulatory region. So the reactivation of the gene, which is atypically methylated, is a potential goal. And those approaches in the 90s tried to reverse this atypical methylation. The drugs that were used targeting the DNA methylases and histone deacetylases were toxic, and the work didn't progress. Now there are approaches that have been entertained more recently, using non-coding RNAs and using the CRISPR system, at least Cas9, for the demethylation of cytosine, which actually is an approach of great interest in oncology to reverse methylation of tumor suppressor genes. But also the talk has been introducing gene editing with CRISPR to try to go to the culprit, the trigger of DNA methylation, is FMR1 methylation, which is the CGG repeat, editing that portion. Even other approaches have been discussed is delivering the open reading frames. And more recently, because of the success of AAV type of vectors delivering genes, there have been research using the same technology, delivering FMRP. Now, there are a number of challenges, and you mentioned the one on delivery, which is always, how do you do it? How do you have a uniform distribution? Ideally IV, but IV has a number of issues in terms of toxicity, in terms of securing the appropriate delivery to the brain, and even developing immunity. Intrathecally will be the ideal situation. But there are other questions. FMRP is not a single protein. There are between 12 and 16 isoforms, and only recently have been shown that the FMRP isoform 1 is actually not the most abundant in brain, but actually is the number 7, those lacking exon 12. So there are challenges that had to do with the genetics of Fragile X, but some of the issues that had to do with general delivery of these vectors and other gene therapies. It sounds promising. It may be a new era, but a lot of work to be done.
There's a question from the audience that asked about reversing deficits. Is it possible to reverse the deficits or simply improve upon, existing deterioration with FMRP gene silencing?
Right.
Or anybody? Yeah.
I think that the issue with FMRP is the same with MECP2, the gene involved in most cases of Rett syndrome, is that FMRP is needed throughout life. So whenever you do gene therapy or you introduce normal FMRP function, it's going to be an impact. You need the FMRP for normal brain development, and you need FMRP for synaptic functioning after synapses have formed. So whether it's a correcting a deficit, it depends how do you find that. I think that there is always an opportunity to make a difference, and this is both with genes and drugs.
Any other thoughts by the panelists on the topic?
I would second what Walter said. I mean, I think conceptually we think about Fragile X as a derailment of brain development, and of course, the earlier we can intervene, the better will be the outcome. But it's also clear that in some ways you can view it as a metabolic brain disease, that we have the opportunity to correct altered physiology acutely. Well, for example, as Mark Gurney was referring to in the case of the PDE4 inhibitors in adult males with Fragile X. So there's a lot of potential to improve the course of the disease, even with late intervention.
Do we have a handle on the timing of intervention? I mean, we kind of globally think that earlier is better, but is there a critical time for any one approach or technology that might be used where it will be most effective? I don't know if there's any thoughts by the panelists on that. Maybe Mark Baer or Mark Green.
Well, I can say that very recent animal studies have shown, I mean, it's quite extraordinary finding that a brief period of treatment around adolescence in the animal models is sufficient to correct cognitive deficits that are then tested many months later. So I'm thinking in particular the work of Peter Kine's lab, but we've also recently completed experiments in my lab showing the same thing. More work needs to be done to pinpoint that critical time window, but it can be fairly brief. So less than a week of treatment in an animal model can be enough to set things in a good way and see benefits many months later.
Okay, any other thoughts?
Yeah, if I may respond as well. I think very generally in terms of patients and their caregivers, children and adolescents are in a learning environment. They're still in school. They have teachers. They're being pressed to learn. Adults, by and large, are not in a learning environment or not in the same type of learning environment. So I think the expectation is that for drugs that might improve cognition, there would be a better response in an environment that's promoting learning and memory. Older patients, it's been described to me that they get in a rut. That doesn't mean when you're an adult, you're not learning. We're all learning today as we're sitting on this panel and listening to each other's contributions. But I think in general, if you were to explore that question with the clinicians or clinical advisors, they would advise that we should be doing studies in children.
Yeah, if I can add, as a clinician, I think that the analogy is early intervention. You start early intervention between ages of 1 and 2 when you notice neurodevelopmental delay, and probably that's the critical window to intervene with any type of therapy. Now, it haven't been in panels of discussion with the FDA about gene therapy, and this is a road that is being paved as we drive. They are aware that gene therapy in neurodevelopmental disorder has to be early. Otherwise, the impact is going to be minimized, and all the balance between risk benefits not going to be favorable. So definitely, we had to do intervention early. When is early enough to have the appropriate balance between safety and efficacy? That's the big question. I think that is the fundamental question for gene therapy, but it's also for drug treatments.
So I like to ask,
I think that there's an area of research looking at the critical window of brain development, which I think is very important here. That obviously happens from birth through to the ages of about 2 or 3. And that critical window of plasticity and brain development has certain triggers to actually close it. One of those could be the FMRP protein. So I think there's a lot of interesting research still to be done as to how FMRP influences the critical window, and particularly how it affects the activity of GABA interneurons, which also seem to be critical to determining plasticity in the early developing brain. So I think certainly part of the research that we've been doing with Takeo Hench at Harvard is understanding how this process happens and how particular ion channels again and how FMRP might be involved. I know that Takeo Hench has been exploring this in the context also of Rett syndrome with some very interesting results.
Well, I want to come back to the translational model, the FMRP1 model, just for a moment. I mean, it's essentially all drugs go through the model, basically. But what have we learned about the model in terms of what it tells us and what it does not tell us? Now they're going back to this question also about timing and intervention. What can we learn from the model, and how can we is there meaningful data that we can get from the model, either with respect to its translatability or timing and intervention? I'd like maybe for Mark to comment on that.
I think the largest challenges with animal models is they're animals. So now that we've cloned a number of different human disease genes, and we can alter them in mice in ways that are similar to the change in humans, we have models now of Alzheimer's, models of ALS, models of Fragile X, models of Huntington disease. We have many more compounds that have cured mice than have ever shown clinical benefit in humans. So one, the challenge is we need to understand how mouse biology is influencing the phenotype. So in the case of Fragile X, we are interested in cyclic AMP, and that derives from an initial observation in patient tissues that there's a change in cyclic AMP. As Dr. Barra was describing, in the mice, there's dysregulated protein synthesis in the brain. A physician scientist at NIMH, Dr. Carolyn Beebe Smith, has tried valiantly to show that there's dysregulated protein synthesis in people with Fragile X, and not so far succeeded. So it's really challenging. We have many more compounds that are active in mice than are showing clinical benefit in humans.
Yeah, Mark Bear, you want to comment at all? And then
well, in defense of the animals, I guess what has impressed me has been the evolutionary conservation of the pathophysiology and treatment response. So we're not talking just mice, we're talking about mice and rats, which are separated by tens of millions of years of evolution, and fruit flies, which even more separation, and zebrafish. So that gives me confidence, frankly, that we have a shared pathophysiology. And with respect to Carolyn Smith's results in humans, that those are heroic studies. And there are certainly human-derived tissues that show altered protein synthesis regulation, including neurons. So I'd say that I wouldn't put too much weight on a negative finding in that regard. My feeling is that the trap that we have fallen into, at least in neuropsychiatry, is trying to map the presentation of a phenotype in a mouse onto a human. And so we can be pointing in the wrong direction in the sense that a pathophysiology expressed in a mice may be expressed completely differently in a human. And so we may be misled in what measurements we choose to take to measure a benefit. So that's where I think is the disconnect. I think the mouse studies give us confidence. We're looking in a good, we're looking at a good mechanism. The question is, how will we detect the benefit by targeting that mechanism? And that's a big challenge.
Yeah. So Neil, have you looked at the mouse at all? All the data that's been accumulated over all these years. Have you been able to look at that to see how you might be able to, leverage that to find these novel drugs?
Yeah. So this is a continuing issue, I think, in many diseases where the model there's no characterization of drugs that have gone from the model, sufficient drugs that have gone from the model through to the patient to link effects. And obviously one way around that is to compare data sets with animals and humans. There's some limitation here because, of course, the ideal tissue to get that data from would be the relevant parts of the brain, and that's a really tricky thing to do. So there is a limitation here, but I think Mark's point really around animal models is something that's true in many diseases, and it's trying to understand which mechanisms you can model in the mouse in the first instance to select the best drugs, but then not to use too much parallelism between that and the patient, because there's a risk you measure the wrong thing in the patient and you miss the key endpoint. So there's a care that you need here, just in general, translating drugs from animal models to humans. And I think the more, one of the gaps we have here is just data in general around that comparison. And obviously there's a lot of great ongoing work that will hopefully fill this, but there is a challenge there.
Okay, thanks. Thank you. I'd like to move on to what can we learn from other diseases in terms of application to the FXS community? So I'd like to start that question with Charles, please.
Yeah, sure. And just before I answer that,
Yes.
just coming back to the question of the animal model, which I think is also relevant when you're thinking about other diseases, because I think, as Neil said there, we haven't just been using animal models for Fragile X, we've been using them across a whole range of diseases, of course, particularly in the CNS, which I think is where some of the issues come in terms of the translatability. So, one of the key things that you also have to make sure you're doing when you're translating from the animal model to the human is making sure that the same concentrations of drug are actually being achieved from one to the other. And I think this is often where we make a mistake. We do a sort of single dose study in a mouse, and then we, of course, can't achieve those concentrations in the human for whatever reason. And then we're surprised when we don't get the same effects. So I think translation of the pharmacokinetics is also an extremely important lesson for us. But coming back to the question of what other disease areas should we be looking at? Well, I think one in particular, which I know the field has paid a lot of attention to, is of course in the areas of epilepsy, in particular genetic epilepsies. So some of the rare genetic epilepsies, which are caused by single gene mutations, but give rise to quite diverse phenotypes and syndromes in pediatric and adult onset conditions, I think can teach us an awful lot. Because there, I think the epilepsy field has had a whole armory of different drugs to throw at these indications. And they're beginning to understand how to use that armory of different drugs and also which ones simply don't work. So for example, in disorders like genetic myoclonic epilepsy, a lot of the standard anticonvulsant drugs simply don't work because they're not targeting the mechanisms behind the myoclonus. And so I think in that case, we've learned an awful lot, again, from the mouse models, but also from just understanding the basic pathophysiology that leads up from the original genetic defect. And I think the way that the epilepsy field is going is to concentrate much more now, not on syndromes linked to genes, but on particular pathophysiologies, and try and treat those pathophysiologies rather than treat specific genetic syndromes. And that, I think, is being seen as being likely to be more successful in the near term.
Moving on to groups that help catalyze discovery and development of new drugs, can you talk about how the advocacy groups have been playing a role in this regard? And I open this floor up to I'll start with Mark Kearney first, and then ask others to chime in.
Thank you, Richard. In the US, the Food and Drug Administration has opened a path for direct communication with patient advocacy groups. It's known as a patient-focused drug development meeting, and each disease is granted one PFDD meeting with FDA. So the National Fragile X Foundation in the US recently held a PFDD in February with FDA. The PFDD is a forum for physician researchers, families, and in some cases patients to directly engage with FDA staff. The outcome of that meeting was quite moving. Anxiety and cognition are recognized and reported by the patients and the families as the two key issues for people with Fragile X. Anxiety is a terrible problem in people with Fragile X. It impacts their ability to go to school, to be in a learning environment, their interactions with their community. As they age and males go through puberty, the anxiety can drive aggression towards the family, and in some cases that may require intervention from outside the home. And then cognition becomes increasingly important if you want to move the patient from a family situation into assisted living. And so FDA has created this path to listen directly to the patients. Unfortunately, we still do not have a defined or an antecedent clinical and regulatory path, since no drug has ever been approved for Fragile X. So with regard to anxiety, how do you, FDA does not understand how to interpret anxiety. It's an internal state. A person with intellectual disability really can't give, or in some cases can't give an answer to the question, are you feeling anxious? I'm not sure that they would understand the question. And FDA at this point in time is uncertain whether caregiver or family reports are relevant. In our case, we have improvement in the NIH toolbox. Cognitive performance directly assessed by a patient is improving. FDA doesn't know the clinical relevance of that. So I think there's a lot of education for the regulators, what are approvable endpoints, and in terms of the design of clinical trials, we need better tools. And all of us that are engaged in this now are in both simultaneously running a trial and trying to validate a novel tool.
So, so we're getting a little short on time, coming up towards the end of our time allocation. So I'd like to start asking a question around where can collaborations improve? Where will we benefit? And are there shared resources that we might think about? So I'd like to pose that question to Mark Baer.
I feel that just to actually return for 1 second about the key role of foundations, because it's relevant to this question, which is that one foundation that's had a big impact on this field, in addition to the National Fragile X Foundation, is FRAXA. And FRAXA organized meetings very early on in the field and tried to attract people to become interested in Fragile X that were critically important in sort of seeding this field. And one of the features of those early meetings was this sense of shared purpose, that even though we all, had our own turf to defend, perhaps, particularly in academia, that was all checked at the door. We were really there to put our heads together and come up with a solution to this problem that is Fragile X. And that's the spirit that I think that, of course, sometimes that gets diluted as more people get involved, but I think that's the spirit that we have. We are all rooting for everyone else's success in this. I think the way to facilitate this in part is to share data as soon as it's credible, to share the data so that we can all learn from other people's trials and errors. And I think we're getting very close. I fully expect to see a success in the not too distant future.
Well, thank you very much for your insights there. Would anybody else like to contribute? And then,
yeah, in terms of collaborative efforts, and even the advocacy group could be a catalyst for this. Is one of the important points that was already mentioned is to develop better outcome measures and biomarkers. This is something that benefits the community. And it could be, again, a collaborative effort, also including academia and industry. A biobank, biosamples and biomarkers are very critical. So there are a number of areas where it could be collaboration, even like other fields in which platform trials have been proposed. Here we see different approaches that tend to target a particular signaling pathway that could be more effective in some patients than others. So there is a lot of room for working together.
hey, thank you. I'd like to just wrap up the discussion then with an outlook and some final thoughts. I'd like to start with Charles.
I'm very optimistic. I think some of the recent clinical trials, the trial that Mark Gurney discussed earlier, I think is incredibly exciting. I think it just opens up the opportunities. It shows us the way as well as to how trials can be done and be successful. And all of this is being done by collaborations. You know, the way that we work is through academic collaborations. We couldn't possibly understand all the complexities of Fragile X and the brain circuitries involved without collaborating very widely with academic groups and other industry groups. So I think I'm excited because I think there's a real opportunity now to sort of push forward. We're seeing signs of success. We're seeing that we can be successful in demonstrating efficacy in these patients, and I think it'll just snowball from here.
Very wonderful, Neil.
Yeah, I share Charles's excitement. I think this is a really nice example of how academic groups, industry, patient advocacy groups can come together and catalyze real progress in a disease. And I think I see the start of a movement here that will eventually find some great drugs, some great cures. So yeah, very exciting time.
Terrific. Mark Gurney?
I'm optimistic. I think multiple drugs are needed to address the different phenotypes in Fragile X. There's not one drug that is going to reverse this condition. So I'm encouraged by the number of therapeutic targets now that are in human clinical trials, and I hope for everyone's success.
Hey, Walter?
Yeah, I share everyone's optimism. For some of us, like Mark and I, who have been in the field for decades, I think that sometimes we see the progress going very slowly, but certainly we're going steadily making progress. We have better tools. We have some success stories, and I think that we need to look at this in a very optimistic way.
Finally, Mark Baer.
I have very little to add other than I share in the optimism of the group. I think we've learned so much over the last 20 years. Now we're applying that knowledge. And I would also agree that I don't think we're going to find a single monotherapy that's going to do the trick. I think we have a lot of interesting ideas and targets that will collectively improve the lives of the individuals affected by Fragile X and their families.
Well, thank you all for your time and your thoughts and your wisdoms and teachings. Mark Baer, Mark Gurney, Walter Kaufman, Neil Thompson, and Charles Lodge. Thank you very much for participating in this webinar.